Okay, hello everybody. We are moving on to lab 10 of the nervous system. All right, so I'm going to try to switch my screen here. Let's see if it's going to work. Probably not. All right, let me go here. All right, we've got that. All right, let's try again. Change my share. It just takes a little bit for it to engage. So I have to kind of toggle back and forth a little bit. And then it should. Of course it doesn't. All right, hold on there. There we go. The fourth time is the best. All right. So let me just make sure that I am recording. I think it's recording. Yep, it's recording. I see the little red dot. Okay, so let's start with the document, the lab that we're going to be filling out, and then we'll move on to the images. So this is the nervous tissue and brain. We looked at some of these models in the lecture class, and some actually I think we looked at in the peripheral nervous system, and then we're looking at it again in the central nervous system. Okay, so we have neurons and neuroglia. These essentially are the structures and sequence of an action potential, and how it goes from, one neuron to another at a synapse. So we have an axon, dendrite, cell body, node of Ranvier, myelin sheath, Schwann cell, axon terminal, and trigger zone, right? And to match them with the functions. So again, the easiest way is always to start with the functions and then match it up with the term. So we will start with A, gaps in the myelin sheath where depolarization occurs. And that form of conduction is called saltatory conduction. Right. So that A goes with 4, nodes of Ranvier. Then we have B, increases conduction rate of an action potential. This is the myelin sheath 5. So B goes with number 5. C, conducts an action potential away from the cell body. That would be an axon. So C goes with axon. D, site of nucleus and metabolism for the neuron. Well, that's always your cell body. E, produces the myelin sheath. That's our friend, the Schwann cell. All right, so E goes with Schwann cell. F, site of action potential generation. That would be 8, our trigger zone or axon hillock. G, receptor region of neuron that produces graded potentials. That goes with 2, dendrite. And I will say technically the cell body, on some of these neurons can do that as well, but we're kind of trying to guide you into the dendrite. Most of the time in the peripheral nervous system, you'll have other cells that are surrounding the cell body and kind of help protect it from being overly stimulated. And then H, that's it, contains synaptic vesicles with neurotransmitters. That would be 7, your axon terminal. Okay, then we move on to impulse transmission. Identify the location on the neuron for saltatory conduction, graded potentials, action potentials. All right, so there's 2 types of conduction. The axons of a neuron are either myelinated or unmyelinated. Right? And the form of nerve conduction velocity that's the fastest occurs when we have saltatory conduction, because that whole process of depolarization, repolarization, hyperpolarization, action potential, I'm getting back to your resting membrane potential, I'm sorry, occurs on naked regions of the axon. And if you have myelin sheath wrapped around the axon and you have very small little unmyelinated areas called the nodes of Ranvier, then the only place this action potential really needs to sort of regenerate itself is at those nodes of Ranvier. And they arrive at the axon terminals much, much, much faster, and it travels around, depending about 100 meters per second. So saltatory conduction occurs at the nodes of Ranvier. Continuous conduction will occur on the naked region of the axon. Right? The models that we look at in lab generally all have myelin sheath on them, right? But continuous conduction would occur on regions of the axon that has no myelin sheath. And really, where graded potentials really have to sort of travel to the axon hillock, and from the axon hillock till it reaches its first myelin sheath. Okay, then we have graded potentials and action potentials. All right, so we talked about that, a lot in lecture class. We talked about what's happening, and how you can tell whether it's a dipole or, whether it's a graded potential or an action potential. And we defined sort of some characteristics about them as well as where they occur. Right? So on dendrites and possibly a cell body, that's where graded potentials occur. Action potentials occur on the axon, either by saltatory conduction or continuous conduction. Okay, then we get to the nervous tissue physiology. And I might have misspoken before, because when I was first looking at the neurons and neuroglia, I knew one of these was about, the events at a synapse, from a presynaptic neuron to a postsynaptic neuron. So I think I misspoke, and I was describing that at the neurons and neuroglia, when in reality, it's here at the nervous tissue. We have our presynaptic membrane, our neurotransmitter, synaptic cleft, neurotransmitter receptor, postsynaptic membrane, and synaptic vesicles. So this is what is occurring. It's similar to a neuromuscular junction. Instead of from a neuron to a muscle cell, we have a neuron to a neuron. So we technically call that a synapse. Okay, so these are in order. The presynaptic membrane, it ultimately releases a neurotransmitter. It crosses the synaptic cleft or gap, number 3. It's going to bind to a neurotransmitter receptor on the postsynaptic membrane, number 5. And then we have synaptic vesicles. Okay. So we have the space between the neuron and the effector membrane. That goes with a synaptic cleft or gap. B, the membrane that contains neurotransmitter receptors. Given this synapse relationship of one neuron to another, that would go with the postsynaptic membrane. Then we have C, the membrane that releases the neurotransmitter into the synaptic cleft or gap. That would be the presynaptic membrane, 1. D, the chemical that carries an impulse across the synaptic gap. That's number 4, your neurotransmitter. I'm sorry, number 2, your neurotransmitter. And then E is the membrane protein where the neurotransmitter binds. And that would be 4, the neurotransmitter receptor. And then F contains a neurotransmitter. That would be 6, your synaptic vesicle. Okay, then we move on to the brain. And this is where we start looking at some of the regions of the brain, and putting some functions associated with them. So we have, again, and we'll identify these regions on the models when we look at the different images, but the ones you have to know are the medulla oblongata, pons, midbrain, which is all part of the brain stem. You have the thalamus and hypothalamus, which are technically part of the diencephalon, your pituitary gland, the cerebellum, the cerebral cortex, and their lobes. So that gets divided into lobes. Remember, the lobes of the brain are based on the names of the bones that are found superficial to them. We have ventricles, which are fluid-filled chambers. Arbor vitae, which is the white myelinated region to the interior of your cerebellum, and your corpus callosum. Okay, so we are going to, again, match up structures, or the functions to the structure. It makes it a little bit easier. So let's take A, relay sensory information to the cortex regarding perception of touch, pressure, pain, and temperature, as well as that performs other functions as well. But you only have so much room in these areas here that allow you to kind of either just match the letter up to the function, but that line here under function represents, the space that you have generally either on a quiz sheet when we're technically taking all these quizzes face-to-face in the lab, and the same amount of room on one of your exam sheets. So there's only so much room, to write your response. And so it gives you a place to kind of synopsize whatever the function is that we provided you. So make sure that it will fit in a space provided on a quiz sheet or an exam sheet. Do it, don't do it, that's entirely up to you. Okay, so A, relay sensory information. A relay station is the thalamus. All right, so that goes with the thalamus. B, aids in controlling breathing and works with the medulla oblongata. That would be the pons. Then we have C, contains choroid plexus, which produces cerebrospinal fluid. That would be the ventricles. D, regulates heartbeat, respiratory rate, vomiting, coughing, and sneezing. That would be the medulla oblongata. Water boy. E, white matter tracts that conduct impulses between the cerebral hemispheres, allowing them to function as a unit, or allows interhemispheric communication. Right. That's E. That goes with the corpus callosum. F, the endocrine gland that secretes hormones that regulate growth, development, metabolism, and homeostasis. Well, there's only one gland listed there, so F goes with your pituitary gland. G, white matter tracts within the cerebellum. That's the arbor vitae, tree of life, it's called in Latin. H, contains reflex centers for startle, reflex, auditory, and visual impulses. That is the midbrain. That's where we find the corpora quadrigemina. I, contains specific centers that interpret sensory impulses, control muscle movement, center for emotions and intelligence. This goes with the cerebral cortex, because those functions are found on particular lobes of the cerebral cortex. Regulates the autonomic nervous system, regulates body temperature, hunger, and thirst. J, that goes with the hypothalamus. And K, regulates posture and balance, coordinates skeletal muscle movement. That is your cerebellum. Okay, moving right along. La, 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 la. Okay, here we have the cerebral cortex functional areas. All right, so when I went through or covered this in lecture, we talked about the cerebrum, and we talked about the lobes, and we said there were certain regions on them that either process certain types of special sense information, or maybe just general body sensation, like the somatosensory region on the postcentral gyrus, or maybe motor areas, found on the precentral gyrus on the frontal lobe, and that there's different functions and regions designated on the cortex, which is the outer 2 millimeters of this crinkly part of the brain, that's associated with certain functions. And so this is what this exercise is for. So what we're going to do is we're going to, theoretically, by this point, when we're talking about these cortex areas, we've had a chance to look at the brain, and we look at the lobes and we go, okay, this is the frontal lobe, and then here's the parietal lobe and the occipital lobe and the temporal lobe. So we've done that. Now what I want to do is go and identify exactly what's found on each lobe. All right. So here, the primary motor area, the lobe it's found on is the frontal lobe. And functionally, it initiates skeletal muscle commands. I generally don't ask the functions, I will admit, but I will ask you maybe, what kind of functional areas are found there on that particular lobe. But we can fill it in as we go anyway. So the primary motor area, is the precentral gyrus on the frontal lobe. And that's where, we have that homunculus, that map of the body. We have it on the somatosensory gyrus as well. But when we want to initiate commands to move the hands, then the hand region, on that primary motor cortex will start to light up, and we will start initiating signals to stimulate certain muscles to contract, and it's going to work with the cerebellum and so forth to execute that particular motor movement. Okay, the primary somatosensory area is found on the parietal lobe, specifically the postcentral gyrus, right? And its primary function is to process general body sensation. Sensory information, like general body sensation. The visual area is found on the occipital lobe. And functionally, it processes and puts meaning to visual stimulation from the eyes. The auditory area is found on the temporal lobe. And not surprisingly, it processes and puts meaning to hearing, auditory stimulation. So sensory information regarding hearing will be received here, and processed. Okay, the gustatory cortex, generally found on the temporal lobe as well, and it receives and puts meaning to information regarding taste. Well, actually, technically, let me go back. The gustatory area, let me go back, is really, we used to, let me just change that. For the gustatory, put parietal to insula. You can put parietal lobe / insula, INSULA, because in lecture, we talked about the insula lobe, and technically, that's really where it's located, but some folks will say, well, it's partially on the temporal lobe, partially on the parietal lobe. And I think for most of your instructors, if you don't have me for lecture, would either be saying more parietal or parietal / insula. So let me just correct myself before we get too far, but it's gustation is taste. Okay, Broca's area is on the frontal lobe. We said it's sort of at the inferior anterior aspect of the primary motor cortex. And it initiates muscles of speech. Remember, we got Broca's speech area and Wernicke's speech area. This is Broca's. The olfactory cortex is found on the temporal lobe, and that receives and processes the sensory information regarding smell. And then the prefrontal cortex is found on the frontal lobe. And this is the area of the brain that's associated with intelligence, judgment, reasoning, all the different variables that are unique in each individual that essentially form their personality and makes them what they are. Not makes them what they are. That's it. Just what makes, you know, what makes them different from other individuals. Okay, then I start to integrate my cranial nerves here, because next week we really identify the sensory and motor functions of the cranial nerves. But I find that if I break it up into 2 labs for you, you end up doing a lot better, because it's a lot of information about cranials to process all at once, you know, in one. So what I ask you to do is, at least using the model, and this picture is the closest picture I can get to what the model actually looks like, you know, in the lab, is to start to memorize your cranial nerves 1 through 12. You should know their Roman numerals, and at least start naming them. There are plenty of mnemonics out there to help you do that, but I would start by just writing 1 through 12 and writing your mnemonic maybe off to the side, and practice olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, hypoglossal, and accessory or spinal accessory. Either is fine. Remember, because most of these are multiple choice or true and false anyway. But then you need to be able to identify these cranial nerves on the models. So number 1 is here, you know, at what's called the olfactory bulb. That represents the olfactory nerve. Blunt cut here are 2 that cross here, what's called the optic chiasm. That represents the 2 optic nerves, or just the optic nerve. Right straight in midline here is your trochlear nerve. Number 3, you know, is actually found oculomotor by pulling the, how can I explain it? By pulling the brain model apart. It's actually, you can see this cranial, a Roman numeral number 3, but you find it on most of the models. It won't be a little nerve root that sticks out here like that trochlear, and then you have the trigeminal next to it. All you'll see is actually, the ocular motor, and then the trochlear next to it. The only one you'll see is the trochlear and not the ocular motor. So hopefully, I'm pretty certain I just zipped you through it in there. There's a model that where the brain is separated, and you can see right at midline, you know, between where you'd find the pituitary gland essentially and the pons here, by the medulla, by the midbrain, you're going to see this little dot, you know, on the inside margin of one hemisphere, or actually it's on both hemispheres, of course, but it's going to be, you know, we'll show you one side, and that represents cranial nerve 3 ocular motor. I mean, yes, it should emanate like that as a little nub, you know, but that is not consistent with what the models look like. If there isn't a picture of that model there, I'm going to make sure there is one. As I go through this now, I'll make sure there is one by the time we have lab on Thursday, because I start here with showing you models of the brain, and, you know, identify this nerve, identify this nerve, identify this nerve. All you have to do is give me the name of the nerve, but if you know the numbers, it becomes pretty easy. And then you just go, okay, this is cranial nerve 1, 2, 3. That's the 4th one. What is cranial nerve 4? And now you've been rehearsing it, so you should be able to tell me that would be trochlear, you know, or the one here at the end, which is your hypoglossal nerve. That's cranial nerve 12. But I would start by just numbering them and go, okay, this is cranial nerve 1, 2, 3 is technically on our model is going to be found to one side or the other, whichever hemisphere you're looking at, just about this region off to the side, and it'll have a little Roman numeral 3 on it. Right, so that's would be the location of 3. Then we have 4 in pink. 5 is trigeminal. It's a big chubba lubba cranial nerve, lateral mid pons. Then we have abducens, which is cranial nerve 6. We have facial, which is cranial nerve 7. We have the vestibulocochlear nerve, which is cranial nerve 8. And once you start with 6, 7, 8, 9, 10. All right, so we have glossopharyngeal, vagus, and spinal accessory. They sort of form an arc from midline going out laterally. So these do follow a certain amount of numerical sequence, but it kind of makes a little bit of an S shape to it. So once you get to midline, right, and just below the pons and superior to the medulla oblongata, you have 6, 7, 8, 9, 10, 11 is multiple roots here out laterally, about anterior and inferior to the cerebellum. And then 12, you come right, like make a circle, and there's a green little area on the area of that model. I call it the putting green. And that's where you find the hypoglossal nerve, cranial nerve 12. So you sort of make a circle from 6, 7, 8, 9, 10, 11, and then 12 is back up on the putting green. So I want you to be able to identify cranial nerve 1, cranial nerve 4, cranial nerve 6, 12, 11, 10, 9, 8, 7, 6. You need to be able to identify which cranial nerves they are and give me the name. I won't necessarily ask you the number, but I start by just counting them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 to make sure I'm identifying each one appropriately. And then use, okay, if that's number 9, then what is number 9? That would be glossopharyngeal. So that's, if we conquer that part now, then next week's lab, when you have to put sensory or motor or both functions to those same cranial nerves, and also be able to look at models that would represent taste, like the taste bud slide, or let's say for accessory spinal nerve, it's motor. Remember I said when we were in lecture, I said the first 2 nerves are sensory only, the last 2 are motor only. Well, spinal accessory is the motor supply to sternocleidomastoid and trapezius. So, next week, we're going to be throwing some models at you, and going, okay, hey, what if I pointed to the trapezius muscle? Now that we've identify that muscle, maybe the origin, insertion, or the action, right now we're going to see like, hey, what cranial nerve, supplies that muscle, or is the motor supply, or, what cranial nerve is associated with trapezius, or maybe pointing to sternocleidomastoid? Then the answer would be spinal accessory. So you need to identify these nerves on this model like this. Ultimately, I could point to one of those cranial nerves and just say, hey, name that cranial nerve, or give me the sensory function of it, or the motor function, if they have both, maybe they just have one. I could point to the optic nerve and say, give me the sensory function. Well, optic is only vision. It doesn't, the first 2 cranial nerves are sensory only. So that's why, it becomes a little more complicated next week, but if we split it up, it becomes much, 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 much easier. And people have a tendency to do much better on their quizzes and exams. Okay, so this might differ a little bit compared to other instructors. I'm pretty certain that most people will do the cranial nerves identification and all their functions next week, but I prefer to do it this way. And I find generally you guys do much better, and let's just start cranking out the memorization part of this. Okay, so that being said, let me stop sharing that because now we've filled that in. And we're going to get to, let me go here and get into the course content so we can look at some images. I do have some other links up here. One is another image of the brain just below the lab. So you can use that to also, and let me just open it up real quick. Let me see how long it takes to actually, this is an older model of let me just switch, I can't find that. Older model of a brain. Back into, there we go. Should be. I hate it when I move this thing and it squishes out my links. Hold on. I think it dropped me out of the Zoom again. So hold on, let me see if I can find, oh, there we go. Okay, hold on. Boy, sometimes it drops me out. It makes me crazy. All right, let me swap this. All right, so let's get to our image here. Takes a while for me to be able to toggle back and forth. So thank you for your patience. I have to keep trying it over and over again so I can see the other image to select as a share. There we go. It took a little bit, but all right. Okay, so this is, it's an image of an older brain model, but it points to things fairly well also. Here we have, and we might have an older, but like one older version like this in the lab, but most of them are a newer version. I believe some of them up in Clearwater, well, the last time I taught up there, they looked like this. Okay, so we have olfactory, optic. You know, you might have a little nub down here. It might just be a spot on one hemisphere, about that same location between, you know, the midbrain and the pons. That would be oculomotor number 3. Trochlear is number 4. Trigeminal is number 5. Now go way to midline. And yes, they are bilaterally. 3 to 5 is the same as, you know, pointing to abducens here on the right-hand side. So you have cranial nerve 6, 7, 8, 9, 10, 11, and 12. So you kind of make a circle from 6 out laterally, count the nubs in a row, so to speak, 6, 7, 8, 9, 10, 11, and then 12, you know, completes the circle. Okay, so that's what that, I uploaded up, you know, that for you guys. Let me close that. Okay, let me go back. And let's get into the images. The links here, remember some of these links here are other videos that I put up for you, you know, that were, again, I made YouTube mini videos, you know, on a lot of this material when we went into quarantine and what was that, self-isolation, or what do we call that now? That's what I call it, self-isolation. Okay, I am going to reshare to the slides or the images for lab 10. If sometimes I use these, sometimes I don't. So histologically, right, I generally don't ask about the motor neuron because I don't think this is a very good picture of a multipolar neuron. Not that it's you know, the instructor's fault that took the picture. It's just hard to get a good one and put it up on the course page. All right, but this would represent a multipolar neuron. I'm not going to use that slide. I'm like certain I don't. Okay, this takes us to those models again, right, where we have a multipolar neuron. And now you can see, you know, this portion here to the left, you know, would actually, if you could take that off, you could attach it to the bottom of number 3, and that would make one big multipolar neuron. So this is the model of it. 1 is pointing to dendrites, 2 is pointing to the cell body, 3 is pointing to your axon hillock. Right? And here we had these other little chocolate kisses attached to it. And especially this one down below, like next to number 3, they cut it open so you can see the interior of that. That would be the axon terminal of another neuron, right? And it even shows you here, that would be that little, I call it the chocolate kiss because it looks like a chocolate kiss model to me, would be a presynaptic membrane. And then the membrane here on the axon terminal or on a dendrite would be the postsynaptic membrane. And you can see little synaptic vesicles in there. Right? Dendrites, cell body, and axon hillock. Right, so the other, while we're there looking at these, and I'll just go back, when it comes to, it was exercise 2, I believe, 2 or 3, about the grade, where on a model might you find graded potentials versus action potentials? Graded potentials would be at things like number 1, dendrites, action potentials down here at 3, and just below 3, where you have the beginning of your axon. Okay, graded potentials on 1, action potentials on 3. Okay, then we have the other region of that model. We represented right through the center here, covered in gray, but cut open here so you can see mitochondria is the axon. The pink cell here with the darker pink-red nucleus is a Schwann cell. And the yellow fatty material wrapped around is the myelin sheath of that Schwann cell. Number 3 is pointing to the little gap between one, myelin sheath of one Schwann cell to another, and that would be your node of Ranvier. And then 4 is pointing to that connective tissue layer that's surrounding one neuron, and that would be your endoneurium. So we have an axon, Schwann cell with its myelin sheath. But if, someone is pointing to just the cell and it says identify the cell, then say Schwann cell. If I'm pointing to, and he missed, the myelin is misspelled here, but if pointing to just the yellow portion, that's the myelin sheath. So you have the cell in the pink, and the myelin sheath is the fatty part. In between those are your nodes of Ranvier, and then 4 is the endoneurium. At those nodes of Ranvier, that's where we have saltatory conduction occur. Very, very fast. All right, this is what I call the chocolate kiss model. All right. It's rather large, and on the other side is going to be that other clear plastic region. So we have on this model, we want to identify the brown exterior layer here, which would be the, membrane. It's the presynaptic membrane of this neuron. 2 is pointing to the white bulbs or balls, which are the synaptic vesicles. And 3, where you see these white balls cut open, it's trying to point to the grainy material inside, which is the neurotransmitter that's stored there. Right, so we have presynaptic membranes, synaptic vesicles, and then the neurotransmitters. Right, so you should know, so we're just identifying structure, right? We're looking at, let me just go back. We have a presynaptic membrane, synaptic vesicles, neurotransmitters. That is, and let me just open up my book so I know exactly which, that was from exercise 3. We have functions associated with these. So, we might, point to number 1 or number 2 or number 3 and say just identify 1 or 2 or 3, or identify the function of 1, identify the function of 2, or choose the function and I give you a multiple choice. Then on the other side of the model, because it's 2 pieces, is this clear plastic region, right? And these black dots on them. This whole thing in the clear plastic represents the postsynaptic membrane. And the black dots represent the receptors for a neurotransmitter. So again, structure, function. The whole clear plastic, number 1 is pointing to the postsynaptic membrane, and number 2 points to the receptors. And that's, they function to bind to a neurotransmitter. So identification of those structures and the functions is coming from exercise 3. Okay, then we start looking at the brain. Right, this is one hemisphere. Right, we have region 1, region 2, region 3. We have the medulla oblongata, the pons, and the midbrain. By the way, while I'm here, right, just follow the arrow of number 3. You can see 67. And then a little to the right, you see that Roman numeral number 3? That I was trying to explain, and not that articulately, is where we would generally, that is the location on these models to identify cranial nerve 3. That's it for ocular motor. It's just this little spot right there above the pons and sort of the anterior aspect of the midbrain. That little Roman numeral number 3 represents ocular motor cranial nerve. While we were there, I just thought I'd point that out because it was quite visible. All right. Then we have above that, right, and to the other side of the hemisphere, we have this, not just a yellow dot there, but that whole oval region, kind of an egg-shaped above and below that yellow dot. All of number 113 is the thalamus. Below that, this little triangular area is the hypothalamus, and then the pituitary gland. Again, structure, function, structure, function. Now we're looking at the cerebellum and the arbor vitae in the center. So the cerebellum is this cauliflower kind of structure to the posterior inferior aspect of the brain. All the crinkly part up there is all cerebrum, but this structure here is the cerebellum, and the white portion in the center is the arbor vitae. 2 points to the corpus callosum that allows interhemispheric communication from one hemisphere of the brain to another. And 3 represents a region on the other side of this like white membrane here. This is where we find one of the ventricles. We find a ventricle, the lateral ventricles to the right and left, inferior to your corpus callosum. The 3rd ventricle is found here between the 2 thalamic adhesions. And then just in front of, like when you follow number 1 arrow between the cerebellum and the medulla oblongata, that little indentation area there, that's where your 4th ventricle is. But you just need to know 1, 2, and 3. 1 is the cerebellum, and the arbor vitae is the white portion within it. You have the corpus callosum, and the ventricles, they contain cerebrospinal fluid. That's where we have the ependymal cells that make this cerebrospinal fluid. Okay, then we're looking at the cerebrum, and this lets you identify or helps you identify some of the different lobes. Right? We have 1, 2, 3, and 4. 1 is pointing to, in the blue gyrus hump there, that is the post-central gyrus. It functions as the primary somatosensory area, and that whole lobe is the parietal lobe. 2 is pointing to the primary motor cortex. From 2 all the way anteriorly, way to the left, that is all frontal lobe. And the groove between 2 and 1 is called the central sulcus. 3 is pointing to the lobe, this sort of deep to your ears, that would be your temporal lobe. And 4 is pointing to the occipital lobe. All right, so we have primary somatosensory area. That's the post-central gyrus as a structure. That's the parietal lobe. And then 2 was the red hump. That's your primary motor area functionally. That is the precentral gyrus on the frontal lobe. We have the temporal lobe and the occipital lobe. Now let me just see what the next picture is. Okay, I don't use those, the sheep brains. So let me just go back. Because some of the other models, and I think I use a different model in the video, the different lobes are color coded so you can see, the different areas. But you should, when it comes to, you should be able to identify that's the cerebrum and what the different lobes are. All right, you have frontal, parietal, temporal, and occipital. If I pull up on that frontal parietal area and pull down on the temporal, that's where the insular lobe is located. But we generally in lab don't ask you to identify because the models won't let you peel all that away. So you just have to know those 4 lobes, but you should know what functional areas are found on those lobes, and that's part of, the exercise, I believe it's 5, where we talked about the functional cortex areas on the lobe and then what those areas function as. I believe it's exercise 5. So here on number 4, I could ask you, identify that lobe, and you would say that's the occipital lobe. What if I asked you, identify the functional area on that lobe? Well, the occipital lobe only has one functional area, and that's your visual cortex, or your primary visual cortex. On number 1, right, that's not only pointing to the postcentral gyrus that functions as a primary somatosensory area, also on that parietal area and somewhat onto that insular region is the gustatory area. Number 2, which is pointing to the primary motor cortex, is that whole lobe is the frontal lobe. We find the primary motor cortex, we find Broca's speech area, and we find that prefrontal lobe that determines personality. So I could point to 2 and I could say identify that lobe, or identify 1 or 2 functional areas on that lobe. And then when we go to the temporal lobe, which is 3 out by your ears, same thing. Identify the lobe, temporal lobe. Identify 1 functional area found there. Well, we have the olfactory area and the auditory area there. So, again, it's got to be multiple choice or true or false. So just know by looking at these what the lobes are and what functional areas might be found there. 1 and 2 are specific: primary somatosensory area and primary motor area, or precentral gyrus and postcentral gyrus. Okay, I don't use the sheep brains. And that pretty much takes us to, that's it. Okay, so again, the cranial nerves are labeled. That shows you pictures of where the cranial nerves are located. I will go try to find another image that would show you the undersurface of the models that would identify the cranial nerves from the models we have in the lab room. I believe I have that in one of these videos, like especially the brain, the last one. I have one for the neuromuscular junction. I have a video on the cerebrum and the lobes, and then I have the brain. And I think that is it. So I'm going to stop there, and we will. I will still live Zoom for a period of time, at the beginning of lab on Thursday, and please, attend one or the other, because attendance is. You should be attending these things on a regular basis. I know some of you, can't, but try to jump in even just for 5 minutes. Okay, so that's the end of the Zoom. I'm here in my office. Take care, and we will see you on Thursday. No quiz due Wednesday night, right? Because we have our exam next week, so don't worry about taking a quiz.